going to walk you through a journey that I've had for the last 5 years, and I hope that will convince you that it was worth starting in the first place. So the umbrella title of my presentation will be Ion Mobility Mass Spectrometry for Isomeric RNA Biomarkers. And my outline, first of all, I'll discuss with you what biomarkers are, what is ion mobility, and then I'll discuss with you, or I will present the challenges that come with ion mobility and what we have done, or a method that we have developed to see whether we can tackle this particular challenge that comes with calibration in ion mobility. And then the third part, I'll discuss the effect that ion mobility has on, or the ion mobility instrument that has on the internal energy of ions, and a kind of project that we have come up with, or a method that we have developed to gauge the internal energy of ions as they traverse the instrument that I'm going to show you in a short while. And then the fourth part, I'm going to discuss the analytical challenges that come with human microRNAs, which are biomarkers for diseases. And then the last part, briefly, I'll discuss about ICOPS, NSF ICOPS, a project that I got engaged in the last few months of my study here. So for the introduction, I always want to start with a quote from Francis Crick. He got the Nobel Prize in 1962 for the discovery of the structure of nucleic acids, specifically DNA. And he said that if you want to study or to understand function, study the structure. So we are interested in understanding how RNA works because it's a biomarker for various diseases. And therefore, we need to study the structure. And the only way that you can study the structure is by developing methods. So the central dogma for molecular biology dictates that the genetic information is written on the DNA. And then this DNA information, or genetic information, is transcribed onto messenger RNA. And then the messenger RNA dictates the synthesis of proteins. And proteins are actually the workhorses for cellular processes. And so the gene, or the DNA, can code different types of RNA. One of these RNAs is microRNAs, and as I shall discuss later, microRNA has been reported as biomarkers for various diseases, and that's why we are interested in characterizing it. And then RNA, in order for it to survive in a milieu, or in a soup of several biological molecules, develops a lot of modifications, or undergoes several modifications, and these modifications can actually be isomeric. So whatever you are seeing here is M6A, or the adenosine, which is a building block for RNA, and it can be methylated at positions either 6 and position 1, which both of them are actually isomeric, M6A and M1A. So the building block for the RNA is actually consists of the phosphate, the phosphodiester bond, which is linked to the sugar, which in the sugar is ribose, and then the sugar is connected to nucleobases. And then these nucleobases can either be adenine, uracil, or adenine, guanine, uracil, and cytosine. And as I talked about Watson and Dr. Crick, heme of Watson-Crick base pairing, we have a uracil base pairing with adenine, and guanine base pairing with cytosine. So it is very important to understand that these basic fundamentals about RNA. So through hydrogen bonding, uracil with adenine, and guanine with cytosine. So we talk about modification, and here I select some few types of modification. So currently, RNA has been reported to undergo like 170 different types of modifications. So these are just selected types of modification. Methylation is one of the most common type of modification, and this modification can happen at different positions on the RNA molecule. For example, it can happen on the ribose. It can also happen on the nucleobase. For example, here I've given an example of adenine or adenosine, and the methylation is happening at position 1, and here at position 2, position 6 and 8. So because we are mass spectrometry based, or we are generating a mass spectrometry-based method, then we are interested in the fact that all these are isomeric or isobaric. So if you shoot these samples, all these samples in the mass spectrometry, then you realize that you get only one mass peak from mass spectrometry. So we are interested because they are actually biomarkers for diseases, as I'll discuss in the next slide. So what we are seeing here, why are we concerned about modifications of RNA? So if you look at this structure of the transfer RNA, if we have the canonical nucleotide, the base pairing, the Watson Crick base pairing, occurs normally. But if there is methylation of A and it becomes M1A, then the secondary structure of the RNA changes, and this has been associated with several diseases, including obesity and diabetes, and that's why we are interested. So why do we want to generate or to develop methods that are mass spectrometry based? It's because of high accuracy. Mass spectrometry, as we know, generates high accurate data, and then high sensitivity. We can go up to attomole or femtomole concentration, and we know that when we talk about biosamples, we do not have a lot of samples to analyze. So sensitivity is a very important issue. So we can also detect, using mass spectrometry, modification that shifts the mass of the analyte. For example, if you're having a nucleotide and then you add a methyl group, that's 15 Daltons, that shift can be detected by mass spectrometry. So we're also using bottom-up kind of sequencing approach, we can be able to detect the modification and then stitch back after fragmenting the RNA. So when you couple mass spectrometry with ion mobility, you're going to get a higher level of information of your analyte. And then there is a potential to resolve isomeric analytes. And that's why we are interested. So in part 2, I'm going to discuss with you how to present what ion mobility mass spectrometry is. And generally, when we talk about ion mobility, what comes in mind traditionally is about drift tube ion mobility. And in drift tube ion mobility, we have the drift tube, and it has an electrostatic electric field, meaning that the electric field here is uniform. And then within this drift tube, there is a buffer gas that is flowing, usually inert, like nitrogen or even helium. So when you do the ionization of your sample, and then it moves through the drift tube, it is separated according to the size, according to the structure, and according to the charge of the molecular ion. And what happens is that, just for example, if you have these two papers, assume that they are isomeric, and they have the same mass, but different structures. So if you have one crumpled and the other one is just elongated, and you drop them, assuming that this is the start of the drift tube and the bottom is the detector, and then in between is actually the buffer gas. Once you drop them, you realize that the crumpled paper arrives fast, and then the elongated one takes time. So what that means is that if you have two isomeric structures and you want to separate them, then the one with which elongated will take longer time, and the one which is compact will take shorter drift time. So that's basically what ion mobility is. So, but then, anytime you want to do a mass spectrometry-based experiment, the workflow involves generating your sample, and then you prepare your sample, and then you have analyte introduction into the mass spectrometry, and then you ionize your sample. And then once you ionize your sample, before you carry out your experiment, of course, you do calibration. And then once you do your calibration, your ions goes to the detector, and then from the detector you can be able to interpret your data. And once you interpret your data, you can use the computer for all this, to control the instrument and to interpret your data. The challenges that come with ion mobility is that there lacks a universal calibrant, which can be used on this platform and on another platform, such that we can have the confidence of comparing data from these two different platforms, or in different laboratories. So we lack a universal calibrant in ion mobility to compare our data confidently. So, and that is the question that we want to answer. Can we use one of the ionization methods that is known to solve part of this problem, if not the whole of it? So there are two types of ionization methods that are well known in mass spectrometry. There are several of them, but two of them are very common. One of it is ESI, which is electrospray ionization, and the other one is MALDI. And MALDI is an acronym for Matrix Assisted Laser Desorption Ionization. So in MALDI, what we have is you co-crystallize your sample with matrices. And a matrix is generally an organic acid that is small in size, but it has conjugated double bonds such that it can absorb a UV laser. And this UV laser is used to ionize your sample. So in MALDI, people shy away from using MALDI as an ionization technique simply because once you use MALDI, you generate what they call matrix cluster ions. And these matrix cluster ions populate between 0 and 1000 m/z spectrum, such that if you want to interpret your sample, you want to interpret your analyte, then it becomes difficult because it can be masked by these matrix cluster ions. So people shy away from using MALDI as an ionization technique. But then, when you couple MALDI as an ionization technique with ion mobility, we realize that you generate a thread line. So what you are seeing here is the blue color is actually the noise, and the intense golden white color is actually the intensity of the peaks. So these are noise, and these are the most intense peaks. So you realize that there is a thread line that's generated once you couple ion mobility with MALDI as an ionization technique. So the kind of instrument that we use is called tri-wave ion mobility mass spectrometry or mass spectrometer, and it's a Synapse G2. And this is the schematic diagram. So what you're having here is the ion source, and it is switchable between ESI and MALDI. So once you generate your ions, they go through a mass filter, which in this case is a quadruple. And then after the quadruple, you go to what they call tri-wave cells. So these tri-wave cells include the trap, the IMS, and then the transfer. So you can do your CID in the trap region, and then you can do separation of your ions in the IMS region. And then you can further do further fragmentation in the transfer cell. So the technology involves having two types of voltage. One voltage is the RF voltage, which is the radio frequency voltage, which keeps the ions within the beam or axial along the beam. And then the other type of voltage is the DC voltage, which is applied on the two electrodes systematically, and it is increased. It's increased after some time. So two electrodes, then there is a DC voltage attached to them, and then it is increased sequentially, and it creates what they call a traveling wave. So this traveling wave travels in the ion mobility cell, and it separates the ions as they serve through the wave. So some of them keep with the wave, and some others remain behind the wave. And that way you are able to separate your isomeric ions. So there are two types of parameters that you can change in traveling wave ion mobility. One of them is the wave velocity, and the other one is the wave height. So remember we said we can generate, so we observe a trend line. So once we change these two parameters, one of them is the wave velocity of 650 meters per second to 1050 meters per second, we realize that there is a shift of the trend line. And then you also realize that this shift happens with any sample that you co-crystallize with. For example, what you're seeing here is the AIP, what is we call auto-inducing peptide, which is just a standard. And it's, the mass is 961, and it's singly charged. So the shifting of the trend line happens with the sample. So we ask ourselves, is it possible then to use this particular method as an internal reference in ion mobility? And then it doesn't matter whether you're ionizing your sample in positive or negative mode, this will still happen. So once we check the wave velocity from 650 to 850, you realize the sample changes the drift time from 8.6 milliseconds to 11.3 milliseconds. And then the trend line remains. And of course, as you can see, we can be able to predict, we can generate an internal reference curve to predict the drift time of the AIP. So again, this happens also as you change the wave height. So for example, at wave height 40 and at wave height 30, the difference is 8.6 at the default and then 14.8 milliseconds once you change the wave height. And as you can see, the thread happens, the thread shift happens with the change, with the shifting of the AIP as the analyte. So what we did is we selected some 5 mass peaks randomly. We just chose them randomly, the ones that are most intense, and then got the drift time for each. And then from there, we got a reference, what we're calling a reference curve. And then we can use this equation to predict with a high accuracy the drift time for the AIP. So what you're seeing here is the curve, and then what you're seeing here is the calculated drift time using the equation, and then the measured drift time. So you go to the experiment and you generate your data, and then you compare the two. And as you can see, the percentage error is actually very low. So we also tried it in a different sample, in this case a polydisperse sample, polydisperse meaning that you have one sample, but then you can generate several mass peaks, and we got the same kind of result. So in summary, for that part, then we think that we can come up with an internal reference using MALDI as an ionization technique, simply because a trend line is observed, and then the shift happens with the sample, and once you change all the parameters or different parameters, the shift can happen with the shifting on the trend line. And the accuracy of the percentage error is very low. So unfortunately, the MALDI ionization source broke down, and we were unable to continue with this part of the experiment. But we believe that we have enough data to submit a paper on this particular project. So part 2, I'm going to discuss or to explain what the kind of method that we developed in order to investigate the effect of the instrument that I have discussed with you, or I've shown you the effect of the tri-wave ion mobility on the internal energy of the ions. So again, to remind you, this is the instrument that we use, and what I've done here, I'm zooming in the ion mobility part of the instrument. So in the ion mobility part of the instrument, we have 2 portions, 2 cells. One of it is the helium cell, and the other one is the IMS cell. So in the helium cell, we have helium gas flowing through at a high pressure. And the role of this is actually to cool the ions, to prevent the fragmentation of the ions as they go into the ion mobility cell. And of course, now the separation of ions happens in the ion mobility cell. You can operate this instrument in 2 modes. One of it is the ion mobility mode, whereby the gases are flowing. And then the other mode is TOF mode, or the ion mobility mode is turned off. And so in this case, the ion mobility cell acts as an ion guide. So just to remember this, such that as we go on, when I talk about TOF mode and ion mobility mode, we'll be able to distinguish between the 2. So when I joined the group, the instrument was relatively new, and we had not understood it, completely, such that, and then there are so many papers that had reported that ions get heated in the ion mobility cell. For example, what I'm showing you are different papers in literature that have reported ions being heated in the ion mobility cell. And the problem with all this is that the temperature that was being reported varied according to the type of ions or samples that were analyzed, according to different groups. So we were at a loss because we did not understand this instrument better. So we decided to come up with a simple method to get the internal energy of the ions as it travels through the ion mobility cell, or the whole platform. So what we did is that you get your ions, you fragment them either in the trap or in the transfer cell. So that is, we are doing that in the trap cell. The green ones are the precursor ions, and the red ones are the fragment ions. And then we can also do that in the transfer cell, whereby you generate you fragment your precursor and generate your fragment ions. And once you do this, you can do this in the 2 modes that I discussed before, either in the TOF mode or in the ion mobility mode. So what you do is that you choose as a molecular ion or a molecule that whose fragmentation pattern is well known and established. For example, in this case, we are having AMP, which is adenosine monophosphate. Remember, it's one of the building blocks for RNA. So it has, for the fragmentation pattern happens, you can get the phosphate, or you can get the phosphate plus the sugar, and then you can also have the base. So you have, these are the fragment ions, and this is the precursor ions. So what happens is that as you fragment it in the CID cell, you will increase the collision energy sequentially until the precursor ions becomes totally depleted. And then you use the intensity of the ions to calculate what we call the percentage dissociation, which is the sum of all the fragment ions intensity, in this case ion count, and then you divide by the summation of the intensity of the remaining precursor ion plus the intensity of all the fragment ions. So once you do that, you get a percentage dissociation. So you increase your collision energy sequentially, and you generate this curve. So this means here that the precursor ions intensity is still very high, and at this point, then the precursor ions has totally been depleted. So why are we using this particular method? It is because, as we know, as the ion travels from the ion source, it contains different types of energies. One of the energies, the kinetic energy, potential energy, translational energy, and different types of energy depending on the degrees of freedom of these molecular ions. So if it has a lot of energy, you require less amount of CID energy in order to fragment it. So if it has some small amount of internal energy, then you require higher amount of collision energy for it to be dissociated. So we chose 3 molecular ions, and in this case, AMP as I discussed, and then another different type of molecule, structurally different, which is berberine, and then the last one was AIP, which is a biopolymer, in this case, a peptide. And we fragmented all of them. So the first thing that we did, remember there are 2 things, or there are 2 parameters that you can change in the tri-wave ion mobility cell. One of it is the wave velocity, and the other one is the wave height. So we varied the wave velocity from 250. So the default wave velocity in our instrument is actually 650. So we increased the wave velocity from 250 all the way to 1050. And what you can see here is that in order for you to acquire 50% dissociation, for example, for 250, you require more collision energy than you will do, for example, at 850. So this means that as you increase the wave velocity, you're also increasing the internal energy of the ions. And therefore, we used this equation, which was reported in literature. It's one of the several papers that reported about the heating of the ions, whereby V is the speed of the ion, Vd is the drift velocity of the ion, which can be calculated by the length of the ion mobility cell. You divide by the time it takes for that ion to traverse that ion mobility cell, and then S is the wave velocity that you are using. So, and we discovered that we are coming up with this red line, which shows you that as you increase the wave velocity, the speed of the ion also increases. So the other group that used this instrument got similar results. I mean, used this equation, got similar results, though their approach was very different and difficult to be applied by different types of research groups. So we think that we have come up with a simpler method to gauge the internal energy of the ions as they traverse through the tri-wave ion mobility instrument. So we also did the wave height, whereby we varied the wave height. And as you can see, as we increase the wave height from 20 to 40, the internal energy of the ion also increases. And this agrees with literature as well. So to remind you again, there are 2 modes that you can operate. One of it is the TOF mode, and the other one is ion mobility mode. So we performed the experiment using the 3 molecular ions, the AMP, and what you can see here is you require more energy in the trap cell to achieve the same percentage dissociation than you do in the transfer, and I'll explain this in a short while. So if you turn it into ion mobility mode, remember in ion mobility mode, there is flow of the gases. You require more energy in the transfer cell to acquire the same percentage dissociation than you would do in the trap region. So this was replicated for all the 3 molecular ions that we used. Remember, we used berberine and then we used AIP. So the story is repeated, whereby here the transfer is on your right side, the trap is on the, the transfer on the left, trap on the right, and then in ion mobility mode, the trap is on the left and the transfer is on the right. And why does this happen? This happens because, like if you can see, this is the TOF mode and this is the ion mobility mode. As the ion travels from the source to the transfer, in ion mobility mode, there are some gases that are flowing into the ion mobility cell, and we believe there is collisional cooling. When there is collisional cooling, it means that the internal energy of the ion gets lower, and therefore you require more energy or more collision energy to dissociate it. But when you turn it into TOF mode, then the ion mobility cell acts as an ion guide. And remember, there are RF voltages, radio frequency voltages, that keep the ions moving. So as the ion travels in the ion mobility cell, it picks up energy, and it is along a distance, like for example from here all the way to the transfer. So it will pick up more voltages or more energies in terms of potential difference than it would if it was traveling just to the trap. So that's the reason why you see the difference between the trap and the transfer. It's because the internal energy of the ions in the trap region is less than the one in the transfer region. So, and then we asked ourselves, what is the role of the gases in the internal energy of the ions? And so the gap that we are seeing between the trap and the transfer in the ion mobility mode. So we turned off these different gases sequentially. For example, you turn off nitrogen or you turn off helium, because you cannot turn both of them off because the transmission will be poor. So in this case, in AMP, we turn the helium off. And once we turn the helium off, you can see you require less amount of collision energy to achieve the same percentage dissociation when the two gases or the helium gas is on. And then in nitrogen, again, we did the same in nitrogen, and with nitrogen gas off, you require less amount of collision energy to achieve the same percentage dissociation. And again, this one agrees with the system that the gases are there and they cause cooling, collision or cooling. So just to rule out, because all other parameters are being kept constant, to rule out the effect of concentration on the internal energy of the ions, we varied the concentration. We used 10 micromolar, but then we varied such that we had the first one, the lowest 4 micromolar, all the way to 500 micromolar. And as you can see from these curves, its concentration of the ions have no effect on the internal energy of the ions. So we believe, again, that we have come up with a simpler method to gauge the internal energy of the ions without first of all, changing or modifying the instrument or using expensive, what they call thermometer ions that cannot be easily found in a simple lab. So we believe that we have proved that you can use this method to gauge the internal energy of the ion as it travels or traversing through this particular instrument. So we did the wave velocity, and then we did both the TOF, or we performed the experiment both in TOF and ion mobility mode, and then we turned off both gases, and then those are the results that we got. So we have submitted this paper, and we believe that it will be accepted. So the fourth project that we engaged in entails about microRNAs. So just to remind you, the central dogma for molecular biology, the genetic information is coded on DNA, transcribed on RNA, and then this is used for synthesis of proteins. Proteins are very important in all in all organisms that are alive. So what happens is that microRNA is encoded by the microRNA gene, and then it undergoes through several processes. several enzymes act on it, like for example, DISA and DROSHA, until these microRNAs becomes mature. And initially they are double-stranded. So as they mature up, the microRNA is single-stranded. So once it matures up and it's like 22 nucleotides long, it lodges itself into what we call RNA-inducing complex. So it gets loaded into RNA-inducing complex. And once it is in this complex, it leads the complex to a target messenger RNA, whereby it binds with the target messenger RNA, and once it binds with the targeted messenger RNA, it causes that messenger RNA to degrade, or it blocks the translation. Remember, translation is whereby you get the protein. So without messenger RNA, you cannot have gene expression. That is why microRNAs are very important. And they play very important roles in gene expression, and they have been reported as biomarkers for various diseases. For example, if we assume that the normal level of microRNA is here, and then you have a disease, the number of microRNAs can either become upregulated or downregulated. And there are several research papers that have reported microRNAs as disease biomarkers for various diseases, including cancer. So this is the distribution of the rank of the microRNA. The most common one is 22 nucleotides long, and because of their size and their small in abundance, if you get a biological sample, the percentage of microRNA is 0.1%. Therefore, this makes analyzing microRNA to be very difficult. So by the time we were publishing this paper, the number of human microRNAs that were reported were 2,500 of them. And it was known that microRNAs are actually similar in structure because they are small. So we are talking about 22 nucleotide long microRNAs. And because they are about 2,500, then we expect there is similarity. But the extent of isomerism was not yet reported. So what we did is that we looked into the literature for each and every microRNA that was reported to be a biomarker for a specific disease. And we found that out of all the reported 2,500 microRNAs, 55% of them were isomeric. And the largest isomer group was 2, meaning that if you have one microRNA, there are 44% chances that there is another microRNA that has the same nucleotide composition but different sequencing. So what that means is that if you're using mass spec, then you are likely to get just one peak, but there are 2 types of isomers in that one peak. Therefore, this offers a challenge in analyzing microRNA. So, and how close is this isomerism? So what you're seeing here are examples, just selected samples of how close they can be. So what you're seeing here, for example, 7A and 7E, the only difference is the red one, G and A. So for this, the flipping of the bases, GA instead of AG, everything else is the same. And the case continues. So similarity, the sequence similarity is very, very high. So why are we concerned? We are concerned because this paper, a group tried to analyze. This is a 21 nucleotide oligo, or 21 nucleotide RNA, we can say. 21 nucleotides, and the only thing that they did was to flip the bases. So instead of having AG, you have GA. Instead of having GU, you have UG. And then they used different types of liquid chromatographic columns to separate. Remember, if you have a sample that contains different types of RNAs, then you need to separate them before you shoot them into the mass spec. So this separation is very critical when you are analyzing a mixture of RNAs. So they found that using different types of columns, that the flipping of the bases will result into coelution of these oligos or of these isomers. So it means that the table that I have shown you of different types of microRNAs that are isomeric, if you are to analyze the 2 of them, it will be difficult for you to use just using mass spectrometry alone, because you will just get one peak from the liquid chromatography. So can you use ion mobility alone? Then this group from Osaka tried to separate or to resolve these two isomeric microRNAs using ion mobility. And what they got, remember, drift time, if you want to have baseline separation of two isomers, then the drift time of the two isomers have to be very different. But what you're seeing here, at charge of -8, they overlap, meaning that they were unable to separate or resolve these isomeric microRNAs. So then we asked ourselves, so because we are talking of coelution of microRNAs, how possible is it for isomeric microRNAs to exist in the same biological sample? So what we did is came up with 3 model diseases. One of it is colorectal cancer, the other one is epithelial ovarian cancer, and the other one is malignant ovarian cancer. And as you can see, remember we said that 55% of all microRNAs pool are isomeric. But when we go into the specific diseases, we realize that 67% in the colorectal cancer are isomeric, and then 75% for epithelial ovarian are isomeric, and even 65% of the epithelial ovarian cancer, malignant ovarian cancer, are actually isomeric. So what this tells you is that the possibility of having a mixture of isomeric microRNA from a biological sample is actually higher than the pool of the human microRNA, as we discussed before. So in summary, for this project, then we have established the extent of isomerism in microRNAs, in the human microRNAs, and since they have been reported as biomarkers for various diseases, then it becomes an analytical problem. And we have also established that these microRNAs can exist in the same biological sample. And this one offers an analytical challenge to all analytical chemists. So the future work for all this work is one of the possibilities is using the instrument to, if you have the two isomers mixed together, you do fragmentation at the trap region, then you separate the fragmented ions and the precursor ions, the fragmented precursor ions, you partially fragment them at the trap, then you separate them at the ion mobility cell, and then you can complete your fragmentation at the transfer region before you analyze using the time of flight mass analyzer. And then from all that information, you can stitch up the information, the drift time and the M/Z information, and try to stitch together and see whether you'll be able to make a call, whether this is isomer microRNA A or this isomer microRNA B. So the last part that I want to discuss is a project that I got engaged in, known as toxicology screening. It is NSF ICOPS, and ICOPS means Innovation Corps. So this project was chosen, and I participated in it. So one of the challenges that we have when we're doing drug toxicology screening is lack of a holistic approach. Usually they follow just one pathway of toxicant. And what we did is, what we propose is that you can use whole cell mass spectrometry to gauge or to detect changes between cells that are exposed to toxicants and cells that are not exposed to toxicants. So like you can see here, the blue represents the cells that are without toxicant, and these are cells that are cultured with toxicant. And then you harvest and wash, and then you use a different type of ionization method, in this case nanodesi, and then you can tell the difference in terms of mass spectral distribution. So what we did is use again the nanodesi, and this is how nanodesi is. It is actually atmospheric ambient pressure ionization. So you have the solvent capillary. So the solvent comes in and it is sprayed on your sample, and then you have a nano spray capillary, and this nano spray capillary is attached to source of voltage, and this enables the ions to move into the mass spectrometry. So what we found is that these are cells treated at different, exposed to the same pH for a different amount of time. So these are the untreated cells. These are 1 hour treated, and these are 4 hours treated. So what you can see is that in the untreated cell, for example, this peak decreases, and this peak is there in the untreated cell, but disappears in the 1 hour treatment and in 4 hour treatment. So, and then we chose for all the experiments that were done, we randomly selected, not randomly, but we selected peaks that disappeared or changed according to the treatment that they were subjected in. So what you're seeing here, red means that they are absent, and green means that the intensity of that peak is very high. So, I want to end my talk with a quote. Today is the 50th anniversary of Martin Luther King, since he was assassinated, by saying that faith is taking the first step, even when you don't see the whole staircase. And then I want to borrow a quote from one of the scientists, Isaac Newton. He said that if I have seen further, it is because I have stood upon the shoulders of giants. And these giants comes in various forms. For example, I want to thank my advisor, Dr. Norman Chiu, my committee members, Dr. Patricia Reggio, Dr. Taylor, Dr. Chet, all the group members, current, and the ones that have left us. For example, Dr. Amanda Watson is here today. Thank you so much for Dr. Chiu's group. And then I want to thank Dr. Chet, Dr. Obaris, and Dr. Todd for providing us with material and technical support. I want to thank Isra for taking her time to put some aesthetic appeal on my presentation. Then the entire chemistry department, and then all this work would not be possible without the support from all those groups. And I want to thank my family back home because they kept me going, so especially my mother, so my family, especially the girls, sometimes we had to come with them here, you know. The Kenyan family, for example, Dorothy Muhoro with her son, and then Dr. Samuel Kimani, who works at PNG, for believing in me and inspiring me. I've made several acquaintances. For example, I used to work at Wellborn Academy during the GK12, when I was a fellow at GK12. So this is a teacher, and then we got to watch football together, and I've had several aha moments like this one when I got some awards at Novoshare conference. I would be glad to answer your questions.
Questions, anyone?
Yes, Dave.
So I was trying to understand, it was like around slide 36 or so. The whole point of that, I think it was like part 2, you were trying to decrease the collision energy. Is that what the point of all those methods were? We were trying to find a method where you have less collision energy for percent dissociation.
Yeah, so the idea is to gauge how the internal energy of the ion changes as it moves from the source all the way to the detector. So we increase the collision energy. The idea is if it has a lot of internal energy, you require less amount of collision energy to dissociate it. If it has a less amount of internal energy, then you require more collision energy to dissociate it. So I don't know whether I've answered your question. Yes, sir.
Do mutations occur on the phosphate head group as well? And if so, can you detect those? Can you talk about the RNA molecule as a whole?
Also, you mean the methylation?
Mm-hmm. Yeah, could it happen on the actual phosphate head group?
I believe it can occur at any place in the RNA, not on the phosphate group, but basically on whatever has been reported is on the sugar and on the base. So I've not come across any modification on the phosphate group. Yeah. Yes, sir.
The last set of mass spec beams that you had for the toxicology experiment that you did were against different conditions of cells. So you pumped into the mass spec the entire cell lysate, everything from the cell.
Yes, so you get the whole cell without further, without further purification or doing anything to the cell.
Proteins or anything, you just put the cell into the mass spec after treating it for an hour or 4 hours.
So you place it on the plate, and then you have nanodist. You spray the solvent and the nanospray capillary is attached to a voltage. So it helps to ionize the surface of the cell. Is that okay?
But you didn't do any kind of filtering from the cell, like you said, specifically target one protein or anything.
No. So these peaks we do not know. So the next project would be to identify what these peaks are, what makes the changes.
Okay.
Yes. Thank you. Yes, Dr. Frost.
Great talk, Joseph.
Thank you.
Fantastic research. So, if Dr. Chu wants to make like $30 billion on patenting this kind of technology, it's different than if you design a new way to make mass specs. Yours is more, designing a method and utilizing the method. Have you guys looked at like patenting that at all or anything like that?
I think because both the ionization technique is open to the public, it becomes difficult to patent such kind of a method. Maybe the approach can be patented. I'm not sure.
Just trying to fund the department here, people.
We can investigate. Any other questions? If not, please join me to thank you.